Viruses are infectious, disease-producing agents which have the ability to enter certain cells and cause disease. Actually, the viruses can cause disease in almost all kinds of living things. They have the ability to enter these normal cells and essentially take over the metabolism, the inside machinery of cells, and convert these normal cells into cells that are producing viruses instead of producing the normal components of a cell. When they do this, they are able to manufacture many, many new enzymes, and these new enzymes participate in the production of these new viruses. In some cases, a cell can, following infection by a virus, can make hundreds of new virus particles. Other cells are able to make, in some cases with other viruses, literally thousands of new viral particles within a single cell. They're able to do this by converting the metabolic machinery from the normal activity into the production of viruses. This involves the production of the new enzymes that we will be talking about today. They use the energy of the cell and the building blocks of the cell in the formation of these new viral particles. Now, outside the cells, viruses appear almost inert, and if you purify them and concentrate them, it's possible in some cases to obtain them in the form of crystals. For example, if you take a crystal of poliomyelitis virus, first obtained in the virus laboratory in Berkeley, and slice through this crystal and look at it by means of the electron microscope, this is what you will see. Now, this has been enlarged about 1 million times. If you would take this picture that you see here and extend it for about 1 mile, the material that would represent in true life actually could be put through the eye of a very tiny needle. So this is a tremendous magnification. Now, if you take one of these little particles that you see here, which represents an individual polio particle, which, by the way, could cause disease in man or in animals, and enlarge it another 65 times, it's possible to get a model such as the one that you see here. Now, this model actually represents about all we know about poliomyelitis. As you can see, it has these protein building blocks on the outside, and inside the structure, you have nucleic acid. This structure is really an amazing structure. It has just exactly 60 of these protein subunits on the exterior. Now, this virus represents a virus that can cause disease in animals or in man. There is another great class of viruses, and that is represented by the bacterial viruses. Here on the floor is a drawing of such a virus. It has a large hexagonal head, and on the interior you see a large amount of nucleic acid. The third great class of viruses is represented by the plant viruses. And here we have a portion of a model of one of these viruses, tobacco mosaic virus. Actually, this is only about 1/10 of the entire structure of one of these viral particles. It's composed of these protein subunits on the exterior, and on the inside we have nucleic acid. Now, it's amazing that all of these diverse-shaped objects have the ability, when they get within certain living cells, to change over the entire inside, the metabolism of these cells, from making ordinary normal proteins and normal nucleic acids, and make these virus structures that you have seen here. This is a perfectly amazing phenomenon. Just one of these particles going into the factory that we call a cell and changing the entire insides of that cell with the formation of new enzymes and new building blocks to make not normal things, but these viral particles. Now, this has been a particular area of interest to one of my colleagues, Arthur Pardee, and I'd like to have him come and tell you something more about this very fascinating subject.
Today I'd like to talk to you a bit about how viruses manage to harm us. You know that viruses can do a great deal of damage. They can make us sick, they can paralyze us, or cause cancers, or kill us. If we knew something about how viruses manage to do these things, we would perhaps be able to prevent some of their actions. Now, the first thing that strikes us about a virus is that it's very small, much smaller than we. We can't even see a virus under a high-powered ordinary microscope. And yet this tiny virus is able to do a great deal of damage. If we think of the way in which a virus acts, we realize that it doesn't attack us all at once. It attacks the fundamental unit of which we are made. This unit is called a cell. We are made of billions of cells. In a picture we have here are some living cells, human cells, which come from the lung tissue. Each of those small white objects is a cell, and each is capable of independent life. Now, what viruses do is to attack these individual cells and kill them. The viruses are still much smaller than these cells, but they are able to damage the individual cells, and when enough of these cells are killed, then the animal itself dies. We remember polio, which attacks only a few cells in our brains, and by doing so can kill. Now, how to study this problem is another question. Many animals are so complicated that we can't study them readily, but we can choose creatures that are themselves single cells, and virus infection is a bit easier with these. In other words, we choose a simpler organism to study than the whole human being. We scientists often do this sort of thing. We choose a simpler model, and it's a little embarrassing because it reminds one of the story of the fellow who lost his keys in the dark and then looked under the light post to find them because there was more light. We always try to look where there's more light also. So we do experiments with bacteria, sometimes called germs, which are themselves single cells of very small size. In the flasks I have here on the desk are some cultures of bacteria. The cloudiness is due to the bacteria. Now, I have two flasks here which are the same, and in here I have some virus. I'm going to add some virus to one of these flasks, and we'll see what happens. Now we'll take these flasks and put them someplace where they are able to let the bacteria grow, and we'll look at them after a little while. We know from electron microscope pictures, such as this one, that the viruses, the small dark objects in this picture, attach themselves to the bacterial cells. Then what happens can be summarized by the animated film that we will look at this point. In this film, we see a cell, and as we come closer to it, we see the wall of the cell, the outer barrier of the cell. And along comes a virus which attacks this bacterial cell. It attaches itself, and then the genetic material, the hereditary material of the virus, is injected into the bacterial cell. Most of the virus stays outside, but what we see is that the genetic material, the dark ribbon, enters the bacterial cell, and nothing seems to happen for some time. There is no virus inside this cell, no infectious virus, and many tests fail to show any change. But suddenly, the genetic material starts to duplicate, and many new models of this genetic material appear. And these are folded up inside new virus particles. Hundreds of these new virus particles are created within each bacterial cell. After a large number of these virus particles have been created, they break the wall of the cell and escape into the medium. This entire process takes about 25 minutes. The bacteria have been killed in this short time, and hundreds of virus particles have been produced. If after 25 minutes we look at the flasks that I infected with virus, we see a striking change. The flask marked with a V, which was infected with virus, has become clear. The flask that was not infected with virus still has the active bacteria inside it. The bacteria have been dissolved by the attack of the viruses. Now, our problem here is to look more closely into the things that went on when the viruses killed the bacteria. To do this, we can't use microscopes because microscopes can't see enough. We have to use chemical methods, methods which involve the chemical changes in the bacterial cells. Now, to talk about the chemistry of viruses, we have to know a little bit about the kinds of chemicals that are in viruses. There are an enormous variety of chemicals in a virus, and we will have time to talk about only a few of these. And in the next movie that you will see, we will examine the changes that occur in some of these chemicals. Here we have symbols of the kinds of chemicals that we will discuss. Now, these symbols in white represent the chemicals that are found in normal, uninfected bacteria. In the first place, we have small particles which represent the food that the bacteria eat. Bacteria have to eat food just as we do in order to grow. Then we see the proteins of the bacteria. Proteins are materials such as are found in meat. Proteins make up the structure of the bacteria. Another very important part of the bacteria are the enzymes, represented by these triangles. Enzymes are the workmen of the bacteria. They carry out the many specialized reactions that are involved in the growth of the bacteria. Perhaps some of you aren't too familiar with enzymes. It may help to know that things like meat tenderizers and junket mixes contain enzymes. Enzymes are found in all living creatures. They carry out a multitude of reactions. Thousands of different reactions are carried out, and each enzyme is a specialized workman who carries out a special kind of reaction. Enzymes are made of protein. They're small, much smaller than viruses, and they are found in great numbers. Perhaps 1 million enzyme molecules are found in a single bacterial cell. They are very important, as you will see in the movies. The last chemical material of the cell that we must discuss is the genetic material. This is the blueprint in the cell that determines whether it will be a bacterial cell or an elephant cell or a corn plant or what have you. This genetic material is very complicated, as can be seen from a model I have here. This model represents a short piece of genetic material. Actually, in a bacterium, this would extend for perhaps a mile. It's a very complicated chemical built of many small pieces. And yet, in this animation, we will see it simply represented by a strip of this kind. This, then, is the genetic material that determines what kind of cell we are going to obtain. This genetic material is built of chemical subunits, which are represented by the small white rectangles next to it. Now, we will see the things that happen in a bacterial cell before it is infected with virus. We see the food particles coming in from the soup in which the bacteria grow. This food particle is changed under the control of the genetic material called DNA into the different proteins and enzymes that are characteristic of the bacteria. This process goes on at incredible speed. Actually, billions of changes of this sort occur every minute in each tiny bacterial cell. These changes result, as I said, into the synthesis of new bacterial parts, including the parts of the bacterial DNA. The DNA is duplicated in the course of about an hour, and when this DNA has duplicated itself, the bacteria divide and one has 2 bacteria where there was 1 before. So, in short, what a bacterial cell does is to take in food and grow as rapidly as possible. Next, we have to ask what happens when a virus attacks a bacterial cell. A virus is a different sort of organism than a bacterium, and what it does is to impose its own growth pattern on the bacterial cell. It shifts the bacterium from making more bacteria to making viruses. Now, We think of a bacterial cell as a sort of factory which is busy turning out more bacterial parts, and we can think of the virus-infected bacteria as a factory which turns out more virus parts. It's almost as if an invading army had attacked a regular factory and had converted it from making something like automobiles into something like making tanks. There are two sorts of processes that go on when viruses infect bacteria. The first is the stopping of the bacterial chemistry, and the second is the starting up of the virus chemistry. Now, to discuss this, we have to say a little about the chemistry of viruses, what they are made of. Now, models, much like those that I showed you a moment ago, except black, we have depicted the parts of the virus, and they are a lot like the parts of the bacteria. We see here a symbol which represents a virus. The virus is composed of an outer coat of proteins, the round black particles, and an inner section which contains the nucleic acid, the genetic material of the virus. And this genetic material, incidentally, is a great deal like the genetic material of the bacteria. Now, the separate virus parts are shown in the symbols next to the virus. The round dots, as I said, represent the special kinds of protein that are found in the virus. The triangles represent special enzymes that are used in producing viruses. These enzymes are not found in the virus particles, but they are necessary for the production of the virus particles. And then we have the strip which represents the genetic material, the DNA of the virus, and we have the little rectangles which represent the parts of the DNA of the virus. Now, let us look at some more animated movies to see what happens in a virus-infected cell. Here's our bacterial cell growing and making its genetic material as it was before, taking in the food and changing it to the various bacterial parts. Now, a virus attaches itself and injects its genetic material into the bacteria. The destructive processes now start. Enzymes are released, new sorts of enzymes, which attach themselves to the DNA, the genetic material of the bacteria, and crumble it into pieces by attacking it chemically. The genetic material has been destroyed, and no new bacterial parts can be created after this point. Instead, virus parts start to be made, enzymes which are important in making the new parts which will eventually appear in viruses. Some of these enzymes actually rob the bacterial enzymes of their food so that the bacterial enzymes can't function anymore. Virus DNA starts up about 6 minutes after the infection, and also a little later, virus proteins are made in the bacteria. These virus proteins and virus DNA molecules are formed for about 12 minutes, and many duplicates of each of these parts are formed. About 200 or so virus DNA parts are made. And as they duplicate, the parts are then surrounded by virus protein parts, and new viruses are formed in this way. This process of new virus formation goes on for another 10 minutes or so, and during this time new enzymes are created which, as we just saw, make holes in the bacterial cell and allow the escape of the viruses. After the viruses escape from the bacterial cell, they are free to infect other cells. In this way, a virus can create new viruses which can attack other cells, and eventually thousands and millions of cells can be killed due to the original infection by only one virus. Whole cultures of bacteria or whole groups of animal cells can be destroyed in this way. The cell, of course, is dead after the virus has attacked it. If we look at an electron microscope picture of a cell, a bacterial cell, after virus infection, we see something of this sort. The bacteria, the large oval objects, are flat and almost empty of their contents. They are unable to carry out any sort of chemical reactions at all and are dead for all purposes. Now, just to recall to our minds all of the things that happened after virus infection, let's look again at the things that take place when a virus infects a bacterial cell. Here's our cell with the virus attached. It injects its viral DNA, its genetic material. This DNA produces enzymes, and these enzymes destroy the bacterial DNA. Now the viral DNA has taken over the operation of the bacteria, and the synthesis of viral enzymes starts, the enzymes that are important in forming the virus components. We see the formation of the new virus DNA at this point, and this continues for some time. Now the synthesis of new viral DNA is almost complete, and we see a replication of the virus DNA. Many duplicates of the virus DNA are made, and the virus proteins, too, start to be made, and the viruses are completed. We have new infectious virus particles in the bacteria, and at this point, the attack on the cell wall begins. The enzymes chew holes in the cell wall, and the viruses escape. So we have seen how one special bacterial virus manages to kill cells and to multiply. As I said earlier, this virus serves as a sort of model to help us understand how other viruses act. We know very little about the actions of many other viruses. We do know that they occur in many different shapes. For example, here is the sort of virus that we've been discussing, a typical bacterial virus. Here is a small spherical virus like polio, and here is a larger spherical virus like flu. We have two viruses that are shaped like long rods, for example, the tobacco mosaic virus. We know also that the chemical compositions of these viruses are different. Some contain DNA as genetic material, like the bacterial virus, and others contain a related substance called RNA, ribonucleic acid. We know a little about where these viruses multiply in the cells, but a lot needs to be learned about this. Also, pictures have been taken of the action of some of these viruses. We know that the cell in the center of the picture is infected with psittacosis virus, and we see it go into violent action as the virus decomposes the cell, contrasted with the cells around the infected one. Now, we can also tell something about the changes brought about by these viruses, the gross changes, and we see very many different sorts of action. With polio, for example, something happens comparable to what happened with the bacterial virus. Polio lyses the cell and the virus escapes. On the other hand, with diseases like flu and mumps, the virus leaks slowly out of the cell, and the cell is not decomposed by the virus. Still, other viruses cause cancers. They don't stop the cell from growing, but rather stimulate its growth, so that cells which normally wouldn't grow at all grow and grow and grow until they kill us. Here is a picture of some minute cancers that have been created by infection with a virus. Each of those little masses of cells show a place where the cells are growing faster than normal under the influence of virus infection. Still, other viruses, for example, the pox virus, kills cells in a local area and causes the appearance of these pustules, which eventually heal to leave deep scars if they don't kill the victim. You can see from a picture of this sort that viruses are nothing to take lightly. We then have the very practical problem of how to prevent virus diseases. And in particular, we'd like to know whether the kind of information that we've been discussing here will help us to cure virus diseases. You know that we can prevent some virus infections even now by using methods of immunity. We can immunize people against smallpox, and we can also immunize them against polio. The other possibility for attacking virus diseases is to use chemotherapy, that is, to use drugs and chemicals which in some way stop the virus infection. We've seen that there are different reactions in virus-infected cells and in normal cells. And if in some way we can stop the special reactions that go on in the virus-infected cells without stopping the growth of the normal cells, then there's a chance of stopping the virus infection. Now, there are two problems involved in this approach. The first is, of course, to find the right chemical, the one that stops only the reaction in the virus-infected cell. And there seems a fair chance that we can find. The other problem is one of stopping the virus infection early enough. We have to have ways of determining when the virus infection starts, and we have to be able to apply the chemical before too many cells are killed. If too many of our cells are killed, it won't help to stop the virus infection because we won't survive in any case. So we need methods of diagnosing virus infections at a very early stage. Or another possibility would be to give everyone the chemical at a time when there's an epidemic, that is, when the virus infection is prevalent. If the chemical is not too dangerous, perhaps we could give a dose to everyone before the virus actually attacks them. So you can see that there are problems and bright prospects in the virus field. There's hope that we will be able to use chemicals and drugs for stopping these dangerous virus infections, much as these chemicals have been used to stop bacterial infections. There's lots of work in the virus field, and in the near future, we hope that we will be able to cure virus diseases and prevent a great deal of misery.
Virus was produced for the National Educational Television and Radio Center by KQED San Francisco, in cooperation with the Virus Laboratory of the University of California at Berkeley. Featured in this program was Dr. Arthur B. Pardee, Associate Professor of Virology and of Biochemistry. The series was made possible by a grant from the National Science Foundation. This is National Educational Television.